Okay, so in the last video, we discussed the types of antigens and the types of antibodies and how antigen and antibodies work together. So to understand the whole immune system, to put the whole picture together, let's understand acquired immunity today. So innate immunity, when we discussed it, we saw that innate immunity works at multiple levels. It works at a physiological level, a cellular level, physical level, but there were a few problems with innate immunity, and those were these four. One is that innate immunity has zero specificity. That is, it works against one and all antigens. It does not have any specific system to work against a specific antigen. So if it is a virus, let's work some way. If it's a bacteria, let's work a different way. Nothing like that. It's one whole system that works against anything that is an antigen, right? Anything that is an antigen, I say, because antigens can generate antibodies in your body. So anything that your body will think of as antigen, it will destroy that. But our acquired immune system has one specificity. So it works against, so it has specific cells for specific antigens, which directly leads us to the point of diversity. So we have a diverse amount of cells, right? So we have all our antibodies, right? We have different kinds of T cells who will work specifically against the antigens to have a more closely knit reaction, a reaction that is made for that particular antigen. It also has a self-non-self discrimination, which is extremely important, because if your body fails to understand what belongs to you, your self antigens, then something called autoimmune diseases will start to form in your body, which still does happen, but autoimmune diseases are diseases in which your body fails to recognize your self antigens, right? So therefore, acquired immunity gives us a huge extent of self non-self discrimination, and the most important is memory. This is why there are many viral and bacterial diseases which we get once and we don't get again in our lifetime. That's because our body knows how to make memory of those diseases, so that the next time the antigen comes, the epitope is recognized and immediately killed. You don't have to go through the whole process of recruiting cells and all of that. No, it's like saying that if, for example, for innate immunity, every time somebody attacks our borders, our government will produce a new line of army, and they will get trained and then go and kill it. But what the country does instead is that it has army trained already. So anytime a type of somebody attacks us, the army is already trained and it goes and destroys the enemy even before it can cause serious damage to our country. It's the similar way, it works in the similar way. So now there are two components via which your acquired immunity works. One is AMIS. AMIS is antibody mediated immune system. It's also known as humoral because it is mostly associated with body fluids, right? So antibody mediated immune system has or works via B lymphocytes. So there are two kinds of lymphocytes in your body, B lymphocytes and T lymphocytes, okay? B lymphocytes are the ones that generate the antibodies, and therefore it is called antibody mediated immune system. So the moment an antigen comes, a B cell will recognize it, present it, and when it will present it, so when an antigen comes, it comes and binds to B cells. The B cells present that epitope, and this leads to rapid division of the B cell into clones. Now what are these clones? These B cells, these active B cells will divide into B plasma and B memory. Plasma B will produce antibodies. So it looks like it does the same thing, right? But plasma B is capable of producing antibodies at a much higher rate, about 2000 ml per dl per minute, right? Very, very high amount of antibody generation. And it will also divide into memory B cells. These memory B cells will become dormant, but they will have a huge lifespan. So what they are going to do is that they are going to go and get sequestered at some point, and when the same antigen comes again, all of this we don't need to do anymore. Immediately, once the presentation has happened up to here, once it has been presented, the cells will not need to divide and form the clones. It will immediately from here, the memory cells will attack and there will be opsonization. Yeah. So this, the division of the activated B lymphocyte into its clones is known as clonal selection. Okay, and obviously clonal selection takes some amount of time. To avoid that, the next time we have the memory B cells. Okay, it's just faster that way. T cell mediated immunity, cell mediated immune system or CMIS, is mediated by T cells. Now, T lymphocytes are basically of 4 types, out of which the cytotoxic T is the one that mediates this immune system, right? So let's just talk about cytotoxic T first. The cytotoxic T, as the name suggests, cyto, cell, toxic, harmful or toxic, right? So cytotoxic T has perforins, secrete perforins, right? Whenever there is an antigen, what it does is that it goes, secretes those perforins. Perforins help in perforation of the plasma membrane or the structure of the antigen. The antigen would leak cytoplasm and it will die, right? But the problem with cytotoxic T is that it has a tendency to be hyperactive, right? So let's say, for example, it really happened a lot during our COVID pandemic. COVID-19 was going and binding to the alveolar surfaces, correct? Once it was binding to the alveolar surfaces, the TC was killing not only the COVID but also the alveoli. Once the alveoli was getting destroyed, there was formation of debris on the lung surface which was causing the pneumonia. So more than the COVID, in that case the TC was harming. That is why the patients were getting prescribed with very high dosage steroids which would suppress the TC. Right? So now, because cytotoxic has a tendency of being hyperactive, it helps in killing cancer cells, it helps in understanding self, non-self much better for our body. But to be able to keep it in check, we have multiple types of other cells. We have the T helpers and the T regulators. T regulators are the suppressors. They are called regulatory cells because they suppress TC and TH. TH on the other hand is a helper, so it helps in recognizing the antigen. MHC is major histocompatibility complex, right? It's protein secreted by our body. It identifies the antigen and helps in the differentiation of B and TC, right? Also helps in the secretion of cytokines. Therefore, it recruits TC, whereas TR suppresses TC. In the same way as B lymphocytes form memory, T also form memory, and they are known as the prime cells or the memory T cells. Okay. Now, when we talk about these cells loosely, one after the other, it's difficult to understand how they work together, right? So let's try and have an example of how they work together. So, when an antigen, I'll write it here. When an antigen enters the system, after having crossed the innate system, okay, and we are only talking about acquired here, the antigen gets presented by different APCs. So APCs help in presenting the antigen. Once the antigens are present, the T helper cells recognize these antigens, presented antigens, and recruit TC and B cells. Okay. These B cells form the plasma and the memory cells, and the plasma cells start secreting antibodies. Now these antibodies as well as these T cytotoxic, they are both specific against this particular antigen and will start killing the antigen. At the same time, your body will also help in making T memory as well as B memory, which will stay in a dormant state for the next attack. So that is what happens when your body is under attack. If in this case the TC is hyperactive, TR will be recruited to control the TC so that it does not end up destroying your body cells. So that is how the whole immune system works. I hope this much was clear. I'll see you in the next video. Thank you.